Hi, my name is Julia. I'm a junior in Columbia College studying biochemistry, and I am a Rabi Scholar. My project was basically looking into changes in RNA processing during stem cell differentiation. So there are two main pathways of RNA processing: alternative splicing, which is like copying and pasting certain parts of the RNA message together, and alternative polydenylation, which is kind of like chopping off the end of the message. It turns out that during stem cell differentiation, the RNAs get longer. It's due to a change in alternative splicing that just ends up producing longer RNAs, and it seems to be pretty important for the like stemness of cells, like it's called their potency. So I was trying to figure out the mechanism by which that occurs, and I thought it might have something to do with the proteins that carry out the reaction. They're called polyadenylation factors. So I decided to look into that this summer using computational methods, and I found out that there were some really significant changes in the versions of the polyadenylation factors that are used during differentiation, which has not yet been described. And that could be a pretty significant reason as to why we are seeing this change in the alternative polyadenylation profile, because if the proteins that carry out the reaction change their behavior because they're the dominant versions of them change, then you will see a change in the resultant products of the reaction they carry out. So that was basically what I did. The implications of this, they're pretty interesting. So, the better we are able to understand something like cleavage and polyadenylation and alternative polyadenylation during differentiation, the better we can understand the process behind stem cell differentiation and why it really occurs. I mean, we have some insight into different transcription factors required for stem cell differentiation and things like that, but overall, we don't completely understand the nuances of why cells differentiate. So this could be one of those kind of unifying principles. This also might have implications in cancer, because cancer cells have alternative polyadenylation profiles that mimic those of stem cells. So if this is the mechanism by which stem cell polyadenylation profiles are modulated, cancer cells also might have this same repertoire of polyadenylation factor versions. And that could be a target for therapeutics, potentially. So those are some of the kind of impacts of this project. Yes, that's pretty much it. I hope you enjoy it. Thank you.